Hey everyone, welcome to our last set of notes in our unit regarding albinism and melanin and DNA and RNA and proteins. So the last part of our story hopefully brings it back to the beginning when we were discussing how it is that an individual can be born albino. And many of you correctly assumed in the beginning it had something to do with DNA, and many of you correctly identified that it's all about mutations. So now that you understand what it is that DNA does, that it's directions and instructions on how to make a particular protein, and you learned how it is you make that protein using messenger RNA and tRNA and the ribosome, let's talk a little bit about when the good things go bad. So basically, what causes albinism is different kinds of mutations that can happen in very particular genes that control the production of melanin. So in other words, if the melanin production is disrupted, you won't make melanin or even make the right type or the right amount, and then a disorder or a disease can result. So let's start with the basics about mutations. Mutations are, they have usually a negative feeling associated with them by most people, but they're not all bad. So we'll talk about some other types of mutations. Mutations are, in general, kind of a good thing for a species. They create variation in a population. So it's why, you, let's say, and other humans have a lot of characteristics in common, but you do also have a lot of things that make you different. So it helps create the variation in the different populations on Earth. A mutation is defined as any change in DNA, the DNA sequence that's in the nucleus. That change can be good and result in, you making a new protein that gives you like an advantage in your environment. For example, maybe it's a protein that helps with digestion. And now because you have this, you can digest something that other individuals cannot. And therefore, that helps you survive in the long run. They can be bad, so they can be things that cause disease, or they can be neutral or indifferent. So they don't harm you and they don't help you, they're just there. We're pretty sure that most mutations are indifferent or neutral. Mutations can also be really big or really small. You can change one single letter in your, let's say, 3 billion nucleotides or 3 billion bases in one cell. You can change one letter, and that can have a terrible effect or a good effect or no effect. You can also change large chunks of DNA, like delete things that should have been there and have missing genes or duplicated genes. So mutations is a word that can mean a lot of things, but it boils down to you're changing something in the DNA. Some mutations will lead to obvious changes in organisms, so you can actually see a change in the phenotype, like your physical appearance or physical trait has changed, like albinism. And then some of them are hidden, so they change proteins that don't result in something that you can see with your eyes. Maybe it's changing the chemistry of your body. So we would call those like hidden mutations. And then, of course, in the negative sense, many people associate mutations with diseases like cancer. Cancer is usually caused by a series of mutations in the wrong place at the wrong time. And then really benign, characteristic about you, like having webbing between your fingers, think like a little bit of webbing, kind of like a duck's foot, or having a mole on your face, something like that. Of course, a non-cancerous mole would be what we'd hope for. Okay, so where do all these mutations come from? Well, there's two types. We have mutations that you do and that you can inherit from your parents. And then you have mutations that you acquire throughout your daily life, just by living in the world. Okay, so some mutations are inherited and some are acquired. Inherited mutations happened in your parents' DNA. So we haven't talked yet about meiosis, but it's this process by which your parents make sperm and egg. So if there are mistakes that happen during that process, and then that sperm or that egg gets fertilized and becomes you in the future, or not you, but a person, then that would be something you inherit from your parents. In the case of albinism was inherited from a recessive allele that a parent or two, both parents in that case, were carriers for. Okay, so it was something that they got. Other mutations can just happen, most mutations are just random. So there's a lot of things that go on in the cell where DNA can just change spontaneously. And there's nothing that you're doing like, oh, I'm doing this lifestyle choice, poorly, and that's why I have this mutation. Yes, there are some things that do cause mutations, but most mutations are probably just random, and because there's so many chances for it to happen. Some of the random times are during DNA replication. So when you want to make more cells, you go through a process called mitosis. But you have to make a copy of DNA for every new cell that you make. And that copying process tends to have a lot of errors. Some of them, most of them get fixed. Sometimes some slip through the cracks. And then those could be considered mutations. When you are transcribing in the nucleus to make RNA for a particular gene, let's say the bases pair incorrectly and like a G accidentally pairs with an A. It doesn't want to, but sometimes it does. And it doesn't get fixed, so now you have the wrong sequence. So you're going to end up like butchering the protein. Something's going to be wrong with that protein. And then there are also things that we do to our bodies that can increase our chances of acquiring mutations. So I'm sure you're familiar with, all the things we know now about cigarettes, radiation from the sun, overexposure to UV light can mutate DNA as well. So there's a whole list of things, it turns out, environmental agents that probably damage DNA, change it, and then therefore cause mutations. So we're going to go through a couple of examples, 3 to be exact, examples of mutations. And I'm going to show you what it looks like, and then you'll eventually have a little practice on there. So some mutations just happen in one single location in anywhere on the double helix. We call this a point mutation, but it's better called a substitution. So a substitution is when you have a nucleotide and it just gets changed to something else. So for example, a T gets changed to a G. Okay, so it doesn't seem like a big deal. It's a small change, but it can have a great effect. But it can also have no effect. So sometimes if you end up changing a nucleotide, but it does not change the amino acid, it can result in no change whatsoever. So it's probably true that a lot of us are walking around with mutations that actually don't have an effect. We call those neutral mutations. But I want to show you some examples of substitutions that can do bad things. Some diseases that you may have heard of, sickle cell anemia, is caused by a change in one base. That happens. Cystic fibrosis, we're pretty sure, is caused by some changes as well to single parts on the nucleotide. So on the picture here, over on the right side, you have, we've just kind of eliminated most of the bases here, but you see you're supposed to have TC, and that's supposed to bond with AG. That's the normal. But let's say that A gets substituted to a C down on the bottom here. Now T is bonded to C. C and G, of course, on the right are still the same. We've changed one base that is going to then change the outcome of our protein. So let's take a look at how an example of this might look. Your job in the future is going to be to transcribe and translate what the protein is supposed to be, and then try to identify what's the mutation that I see here, and then how's it going to change the outcome. So up top here is the completed normal situation. So let's say that you have a gene, and then, here's a very short sequence of DNA starting with TAC. If you transcribe it correctly, you get the second row, and then if you decode what the message means during translation, you build this amino acid chain. Methionine, is that glycine? No, it's glutamine, it's one of those. Valine, and then your stop codon. And of course, in reality, they're much longer. Okay, but let's say that there's a substitution somewhere. So the first step is to identify where the substitution is. So what I want to do is compare my mutated sequence to my normal original. So I'm just kind of spot checking like a word search, looking to make sure that all the bases are the same. And it turns out that they're not. Right here, this C should have been an A. Okay, that was the normal, but it has been substituted in one particular point with a C. Notice that the remainder is all the same. Okay, so this CATC at the end is normal. So now when you transcribe this sequence, the first amino acid is the same, the second one is the same, but the third one transcribes to GGG, which is a totally different amino acid. Now, that's glycine. There it is. And then notice our stop codon is the same. So what we have done is we have changed one amino acid, okay? Now, what the amino acids do is they bond together in a particular pattern. So what makes the protein unique is the way the amino acids bond, and that will actually create a shape in three-dimensional space. But if you change one of the amino acids, you change the shape of that protein. And sometimes that shape can determine its function. And maybe, like in the case of melanin, the protein isn't the same anymore, so therefore it doesn't have the same job. And in this case, or in the case of melanin, it would be to produce this pigment. But the pigment you're producing is not the right one, or it's not even functioning at all, so therefore it's not going to do the same job. So that could be one example of what could lead to something like melanin or, any disease or disorder. Sounds not so bad, just changing one amino acid, but it changes the entire shape of the protein and can make the protein non-functioning. So here's another example of a substitution. It's the exact same problem at the top, so all of this is the same. Here's our amino acid sequence, methionine, GLN, VAL, and stop. And we have a new mutation, a different substitution this time. So let's compare our normal sequence to our mutated. TAC, those both match. GTC, GTC, those match. Then we have CAC, but now we have CAA. So what we see here is in the third part of your codon, you're going to have a change. It's now CAA. Notice that ATC and ATC are the same. Okay, so now when I go to translate and figure out which amino acids we have methionine and GLN match. And notice valine is still valine. And then our stop codon is still our stop codon. Because what you can see over here, let me change my color, is that valine has multiple codons. So sometimes if a mutation happens, you still might end up making the correct amino acid just by chance, because many amino acids have more than one codon. Okay, so our result here is there was a mutation, but you wouldn't even know it because there's been no change in the amino acid sequence, which means the protein is going to be totally normal and everything is business as usual. So we would call that a neutral mutation. Okay, so substitutions can be terrible, they can also be neutral, or they can be good. So maybe having a substitution changes a protein shape and makes it better or makes it more efficient. We wouldn't actually know that until we were in the situation. And I don't think everyone's going around all day trying to figure out what mutations everybody has. But we know and we assume that many of them can actually benefit you. Okay, so let's go on to a new type of mutation. Some mutations we identify as deletions, and they're very easy to understand because it's exactly what it says. One of the nucleotides has been deleted. What will happen in real life is that all the other nucleotides will shift over to fill in the blank space. So notice over here, you've got your missing nucleotide. What will happen is everybody down the line here will shift over one position to fill in the blank. And now what you've done is you've changed all the codons. So all the codons are off by one, which means you're going to change a lot of amino acids, and you are greatly going to affect the protein being made. Probably the protein isn't going to be made at all. Muscular dystrophy is an example of a disease that's caused by a deletion mutation in a very particular location on a chromosome in your DNA. So it has to happen in this one particular spot and affect this one particular protein. So here's what that deletion might look like. So once again, starting at the same, we've got our sequence of DNA. It's the same one we've been working with, our normal at the top. Now we have a mutation. So this is a little trickier for students to see sometimes. So I'm going to show you a little trick. If you were asked to figure out what kind of mutation is this a substitution or a deletion? What you want to do is just start comparing your sequences. Don't necessarily compare the amino acids because, remember, some mutations can still lead to the same amino acid. So always look at the DNA. So we have TAC is the same. We're supposed to see GTC, but what we see instead is GCC. So what is missing? The T. The T has been deleted. It's not there anymore. What has also happened is that all of the other bases have shifted over one position to the left. So how do I know that that's happening? Well, there's a couple of giveaways. One giveaway is, notice there's a missing third base at the very end. So the stop codon isn't even there in the end. And what you can also see is that up top here, we had CAC as our next sequence. And the CAC is still there, but it's just off by a position. We have our ATC, but ATC is off by one position as well. So our codons are now officially different. So when you decode this message here and look up the new codons you have, you're going to make different amino acids, and notice the absence of the stop codon. So this protein would be incomplete. It would continue to code into the next gene, which means you're going to make some jacked up protein if you make one at all, and it will probably be non-functioning. So we call deletions, they result in what's called a frame shift, because all of the amino acids shift over one position, and your protein becomes incomplete. There's also no stop codon. That's a problem too. So how does you don't know when the end of the gene is supposed to be. These are almost always negative mutations simply because you can't even think of a scenario that would make that situation good. So all deletions are going to be negative. Okay, there's one more example here, and it's the opposite of a deletion. It's what if I have a base in there that does not belong? They call that an insertion. An extra base is added. It has a very similar effect. It's going to cause a frame shift, but this time the shift is going to be in the opposite direction. You're going to get bumped downstream or towards the end of the gene. You're going to shift your codons again, and problems arise. We're pretty sure that Crohn's disease is another disorder that's probably caused by insertions, probably amongst other mutations as well. Okay, so our final example here has once again our normal sequence as it should be. And if you were asked to figure out, this mutated DNA, what kind of mutation does it have? You would want to start by comparing the normal TAC, that's a match. We're supposed to have GTC, but what we have is GGT. This looks odd. I'm going to keep investigating. CAC, CCA, something is off. And then what I can also notice at the end here, you've got a base hanging. So everybody got bumped to the right. And that C up here is now bumped out of that codon. Okay, so what is the base that was added? So we were supposed to have GTCCA. We have GGTCCA. So there's the CCA. It's still there. It just got bumped. One of these G's, I'll just pick the second one here, does not belong. Okay, so there's our insertion. It's our extra codon, our extra nucleotide that didn't belong. So that changed our amino acids. Notice once again the absence of a stop codon. So the result is going to be really similar to deletion. So there's going to be a shift of all the amino acids, a frame shift. This time in the other direction to the right. It's still going to change all the amino acids after the insertion happened. It's an incomplete protein. There's no stop codon. The protein probably doesn't work at all, and therefore we consider this a negative mutation. Okay, so those are our three kinds. So what you're going to get is some practice on identifying is this a substitution, insertion, or deletion? And what kind of effect do you think it has on the protein? Do you think the protein's going to be normal? Do you think it's going to be altered? Do you think it's going to be incomplete? So I've got a little summary here for you to take a peek at. I'll send the notes to you as well. But I hope that helps everybody. And that's it. So if you have any questions on mutations, let me know. But there will be practice to come. Have a good one.